All right.
So
Go ahead.
thank you everybody for being here and for having me. I'm Andrea Luppi from the University of Cambridge, and I'll be talking to you about the research that I've been doing in the Cognition and Consciousness Imaging Group at Cambridge, which is about mapping the landscape of consciousness in the dynamics of the human brain. And as this talk goes, I'll try to make each of these parts of this title clear. So let's get started. And I think it's fair to say that what we're interested in the lab, and perhaps myself in particularly, is the flights and perchings of consciousness, or the so-called stream of consciousness, as William James famously described it in the 1800s. And this might seem almost paradoxical because we know that consciousness is precisely something that is ever changing. So how can we ever fix it enough and be able to study it? But I think that this is not so impossible after all. And as this image beautifully shows, there are in nature, and not only in nature, but also in our environment, a large number of phenomena that are emergent, in a sense that they are observed arising from the interactions of lower level components. And we think that maybe consciousness can also be seen as this kind of an emergent phenomenon arising from the interactions between neurons in a brain that then combine to create the experiences that we have. And perhaps this might be what you're thinking at this point, which is that what I've just described to you is not actually science. That sounds a lot like magic. And part of what I'm hoping you will take away from this lecture is that we now actually have the tools to bring this closer to science than to magic. And in particular, the neuroscience of consciousness has been doing over the last 20, 25 years some really great progress. Specifically, we start from a philosophical question, which is the one that I have been just describing, which is how does mind arise from matter? And philosophers have been describing and tackling this question for thousands of years. But we try to turn it into a scientific question, which is how does consciousness arise from the brain? And the reason for this rephrasing is because it lets us take a scientific approach to addressing this question, which is we want to look at brain changes to identify what is different when consciousness is lost, which we can do, for instance, through the kind of neuroimaging tools that have been developed over the last quarter of a century, such as functional MRI. So I will be talking mostly, in fact, about functional MRI, but I should note at this point that many other ways exist of looking at brain signals, and this is just one. And at this point, this scientific approach to the study of consciousness already comes with what you might consider a problem, which is that when I said we want to study the brain and see what changes when consciousness is lost, well, that is in some sense hiding some of the complexity of this question, because we know that there are many ways to lose consciousness. For instance, each of us loses consciousness daily when we fall asleep, and then we regain consciousness the following morning. That is a natural, spontaneous process. However, there are also ways of inducing loss of consciousness. This is something desirable that we do in the context of surgery. We anesthetize patients so that they won't suffer during surgery, so that they can reap the benefits of surgery without the associated pain. However, loss of consciousness can also be the result of severe brain injury. Some people suffer accidents that damage their brain, and as a result, they fall into persistent chronic disorders of consciousness, such as the vegetative state, also known as the unresponsive wakefulness syndrome, or the minimally conscious state. And these are also cases of induced loss of consciousness, just like anesthesia. But of course, one of them is pharmacological, whereas the other actually involves changes in the way that the brain anatomy is organized. And in particular, throughout this talk, I will be focusing on these two cases of induced loss of consciousness. So anesthesia on the one hand and brain injury on the other. So excluding sleep for now. And what we have been doing is trying to identify neural correlates of induced unconsciousness. Now, what this picture here is showing is part of the problem that we face, which is to say that in some sense, we expect the brain function, specifically of anesthetized volunteers, and the brain function of brain injured patients to resemble each other, because although by very different routes, both of those kinds of people have become unconscious. So the behavioral endpoint is the same in terms of loss of responsiveness. On the other hand, however, there are going to be many changes that are not shared between those two conditions. Some patients, as I mentioned, they actually have brain damage. Anesthetized people actually have some particular drug in their system that is unlikely to be targeting just the parts of the brain that are involved with consciousness, but is probably acting throughout the rest of the brain. So there are some dissociable aspects that pertain only to anesthesia or only to disorders of consciousness, and we are seeking instead the convergence between them. Now, this is work that we carried out a couple of years back now. This was the first paper that came out of my work in the cognition and consciousness imaging group. And what we did in this work is we were looking for signatures of consciousness using this approach that I have just described, with the idea being what is common in the brains of unconscious individuals regardless of how they lose consciousness. And I will briefly describe here what we found, which is summarized in this picture. So we were looking at two different aspects of brain function. So our participants and our patients, they were all scanned in an fMRI scanner, which records their spontaneous brain activity over time. And in blue, you can see this quantity named ICC, which refers to how well connected a region of the brain is to the rest of the brain in terms of exchanging information. And on the bottom in yellow, what you can see is instead the entropy of the spontaneous brain signals, which is to say how diverse is this signal. Is this a very stereotyped pattern, or instead is it a very diverse pattern that keeps changing over time? And in both of these cases, we compared the unconscious people, so the anesthetized volunteers or the patients with disorders of consciousness, we compared them with healthy volunteers who were awake before being anesthetized. And what we found is that there were some reductions both in this measure of connectivity, the ICC, which stands for intrinsic connectivity contrast, and also in this measure of entropy. So we found that both in anesthesia and in disorders of consciousness, the unconscious brain loses both connectivity and diversity of its spontaneous activity. This is happening in both cases of loss of consciousness. However, what is really interesting, which is shown in red, is that these losses of information and of connection are not randomly distributed in the brain, but rather they overlap with each other, not just within a specific way of losing consciousness, but also across different ways of losing consciousness. And in particular, as you can see here in the center of this, there are regions of the brain that lose complexity or diversity of their signal and lose connectivity regardless of how someone became unconscious, whether that was due to brain injury or whether that was due to anesthesia. So this is, if you will, a signature of consciousness in this brain function that we observe. However, there is one aspect that, even after this study, was still remaining mysterious to us, which is to say that the various different mental states that each of us goes through during our day, even minute to minute, such as working, sleeping, thinking, and perhaps doing sports and everything else that you can imagine. All of those things are mental states that are supported by one and the same brain structure, because on a first approximation, the structure, the organization physically of your brain does not change over the course of a single day, perhaps just a little. It doesn't certainly change over the course of a few minutes. And yet, over the course of the same minute, you can think among all sorts of different things. So how is that possible? In other words, how does brain function, brain dynamics change, even though brain structure remains the same? And of course, the reason we are interested in this is because we know that for each of those mental states, there must be a corresponding brain state that is supporting it and that is enabling that particular subjective experience to actually take place. And for that, I will show this video. So, as I hope you've seen here, there is this idea that the same physical structure, in this case, a metal plate, can support different and quite varied patterns that are taking place over it. In this case, these are patterns made by the sand over the same oscillating metal plate. This is called cymatics, and it is a well-studied phenomenon. And shown here are just some of the most interesting patterns that you can observe depending on the frequency, specifically at which the plate is oscillating. Now, we think that this might be the way that also the brain works. And why do we think that? We think that because this is not something that just happens with sand on metal plates, which would be fascinating, but not particularly insightful. But rather, it turns out that this is a much more widespread phenomenon. And in particular, it is even the way that we as humans use to make music. As you can see in this picture here, there are different patterns of, in this case, vibration that are happening over the body of a guitar. And this is even more widespread in the physical world, where even the way in which atoms behave actually follows some of these same principles. So these are very fundamental phenomena. And so the question became, perhaps we can replace this metal plate with the structure of the human brain, and instead of grains of sand over it, we can have information spreading over it. And the structure of the human brain, in terms of the white matter fibers that connect different regions, is known as the connectome, term coined by Olaf Sporns and his colleagues. And so the idea is we can use the connectome and understand how brain information spreads over it, and use the same mathematics of the sand over the metal plate to understand what is happening here. And I should clarify that this was not my own idea. In this case, this was an idea developed by Selen Atasoy and her colleagues. But you can see the results of that here. And I would say it's quite striking that these patterns, although in one case they are patterns of sand over a metal plate, and in the other case they are patterns of information spreading over the human brain, nevertheless, there is some fundamental similarity between them. And this is not just because they have the same underlying mathematics, but because they in some sense represent the same underlying phenomenon, which is of diffusion over a medium. And so we took this approach as a way to identify what is happening on the brain, in terms of brain function, which you can think of it as effectively decomposing the ingredients of your smoothie. You have a smoothie, and you might want to know how much of each of these ingredients went into that particular smoothie. So maybe this very red smoothie had a lot of watermelon and strawberry in it, whereas the orange smoothie had less watermelon and less strawberry, and instead it has a lot of mango and orange, for instance. This is the same principle, but we apply it to human brain functional activity instead, to identify which of these particular patterns of oscillation, if you will, was contributing more or was contributing less to brain activity at a particular point in time. And this is called connectome harmonic decomposition. And what you will see here is its application by Atasoy and others, as the researchers who I was mentioning earlier. And they used it in the context of LSD, which I think is an especially fascinating case study, not least because this is a talk that I'm giving at APRA. So hopefully this is something that you will be familiar and interested with. And in particular, what you can see here is that there are relevant differences between the decomposition, the connectome harmonic decomposition, as this is called, that is observed when participants are under the effects of LSD or under the effects of a placebo. This is shown even better here, which is to say that there is this characteristic signature of LSD in terms of brain harmonics. Now, one thing that is worth emphasizing here is the meaning of the horizontal axis on this plot, which is to say, what does space represent here? At the very left of the plot, you see what are known as low frequency harmonics. These are very large scale patterns of vibration. Towards the very right of the plot, you see what are known as high frequency harmonics. These are very fine grained patterns of information spreading over the brain. And this will become relevant later. But for now, just bear in mind that the difference is low frequencies on one end and high frequency harmonics on the other. And you can see quite clearly here that the reason why they are relevant is because they differentiate between LSD and placebo. Specifically, when you are under the effects of LSD, your low frequency harmonics will contribute less to brain function, and your high frequency harmonics will contribute more to brain function. And at this point, you might wonder, is this just specific to LSD? Or is this actually capturing the psychedelic experience itself? And of course, this is also what they were wondering, because a year later, they published this additional study, which was looking at the same analysis, but when their participants were under the effects of psilocybin, so-called magic mushrooms, which is a different psychedelic that uses some of the same neuropharmacological principles because it is a so-called serotonergic psychedelic. And what you can see here is that the signature of psilocybin in the human brain is pretty much identical to the signature of LSD in the human brain. And we were very excited about this. And I should clarify that I was not involved with either of those two studies. But I got very excited about them. I thought that this might be able to tell us something not just about the psychedelic experience, but also about consciousness itself, which is what we study in the lab. So the first question that we asked is this specific to the molecular signatures of LSD and psilocybin, which are both agonists of the serotonin 2A receptor? So basically, you can think of this question as, are we just observing a signature of some specific receptor being activated? Or are we instead observing a signature of the mental states that are induced by the activation of this receptor? How can we test it? Well, we can test it by using a drug that is also considered a psychedelic, but it is a non-classic psychedelic because it doesn't influence this particular receptor, namely ketamine. So ketamine is interesting because at high levels, it is actually an anesthetic. But at sub-anesthetic doses, it can induce psychedelic-like experiences. And so what we found is that actually its signature in terms of brain harmonics resembles the signature of LSD and psilocybin, specifically with an increase in the contribution of these high-frequency hormones. So even though the molecular mechanisms are very different, the mental states that they induce are similar, and so are their connectome harmonic signatures. And at this point, this was important for us as scientists because it was a demonstration that what we were looking at was not just the signature of a molecule, but rather it was the signature of a particular kind of mental state, namely the psychedelic state. And so our question next was, what are we going to see when instead we look at a different way of perturbing consciousness? Not just bringing up psychedelic experience, but actually temporarily suppressing consciousness through anesthesia. So we use the anesthetic drug Propofol, which is an intravenous anesthetic, very commonly used. And what we found is exactly the opposite. That is to say, when we compared our participants before the anesthesia and while they were anesthetized, what we found, as you can see here, is that the signature is exactly the opposite of the psychedelic signature. The low frequency harmonics, which are reduced when you are under the effects of a psychedelic, instead they increase their contribution when you are anesthetized. And the high frequency harmonics, which psychedelics tend to enhance, are instead suppressed by the effects of anesthesia. And after that, we asked the same question that we had asked before, which is this specific just to this particular drug? Because as you might know, there are very many anesthetics, and some of them act on similar molecular mechanisms, and some of them act on very different molecular mechanisms. But importantly, anesthesia, as I was mentioning earlier, is not even the only way that consciousness can be suppressed. For instance, someone can have a disorder of consciousness, where brain damage results in chronic loss of consciousness, whereas anesthesia, thankfully, is only temporary. And so we scanned 22 patients, and what we found, once again, was the same signature. In particular, with the high frequency harmonics being suppressed, just like they are when it comes to anesthesia. And the results of this study, with a lot of other analysis, are currently available as a preprint on bioRxiv, so I would invite you to check that out. But in the interest of time, let's move on a little bit and say, what exactly is happening here? What are we seeing? And to a first approximation, I would say that we have found at least some of these neural correlates of induced unconsciousness in terms of a particular signature of connectome harmonics, with increased contribution of the low frequency harmonics and decreased contribution of the high frequency harmonics. And actually in that paper, we even show that this signature can distinguish between two different subgroups of patients with disorders of consciousness. Those who can imagine playing tennis in the scanner and actually show evidence that they are in fact conscious, even though they are behaviorally unresponsive, versus those who cannot. And so we cannot presume that they are conscious. However, we can see that the other important aspect that comes out of this work is that the signature of anesthesia and the signature of the psychedelic state are, in some sense, the mirror reverse patterns of each other. And this is something that we found very interesting. And in particular, as you can see here, this is also borne out if you use a different approach to study this question, which is to say, we have all of these different comparisons across states of consciousness. We can compare, as I was mentioning earlier, different subgroups of patients with disorders of consciousness. We can compare different levels of anesthesia. What happens when someone is only mildly anesthetized? So they're a bit groggy, but still conscious. And in that case, as you can see on this plot here, their pattern of brain activity is not significantly different from the pattern of a conscious brain. On the other hand, under the effects of ketamine or LSD, there is an increase of diversity in the pattern of these connectome harmonics. So this is a different way to say the same thing, which is that these connectome harmonic signature have two characteristics that we consider important. The first is that they're able to distinguish different states of consciousness. They are able to distinguish between someone who is just mildly anesthetized but actually awake versus someone who is actually deeply anesthetized. They're able to distinguish between patients who can play the tennis task, and so they can give neuroimaging evidence of actually being conscious, versus patients who cannot do that. On the other hand, they also distinguish between perturbations of consciousness that result in loss of consciousness from perturbations of consciousness that are actually of a psychedelic nature, and so instead lead to a higher diversity of your subjective experiences. So we think of this as a method for mapping the landscape of consciousness, if you will. But at this point, you might have one more question, which we had as well, which is, how do we read this map? Now, we know that the map seems to be there, but what does it mean that there are higher contributions of low frequency harmonics and lower contributions of high frequency harmonics? I've been mentioning that quite a few times, and there is a reason. But what is that reason? And it turns out that low frequency harmonics can be identified as those harmonics that are structurally coupled, whereas high frequency harmonics are so-called structurally decoupled. So the structure function coupling is important because we know, as I was mentioning earlier, that all the different kinds of brain function that we entertain during the course of our lives, they depend on the same brain structure, same brain organization. However, I acknowledge that at this stage, these are somewhat abstract words. And to try and bring it a little further in terms of what we understand of them so far, we know already that from some recent studies that when people are unconscious, when they are anesthetized, and not just people, even macaques, when they are anesthetized, their brain function looks more similar to their brain structure. That is to say, if you look at the way that the brain exchanges information, that will look a lot like the way that the brain is actually structurally wired. Now, this might seem surprising, but something that is even more surprising is that the opposite occurs when you are under the effects of psychedelics, like LSD. In that case, we recently showed this very year that while you are under the effects of LSD, the way that the brain regions in your brain exchange information looks significantly less like the way that the brain is actually structurally organized. And the fact that these signatures of consciousness pertain to the high and the low frequency harmonics, now we think makes sense. Because what we are showing is that during loss of consciousness, when we know that brain structure and brain function are more closely coupled together, that is when we see an increased contribution of the low frequency harmonics, which are precisely those that signal higher contribution of structure to function. Vice versa, what we find is that when people are under the effects of a psychedelic, there is lower contribution of structure to function. That is to say, higher contribution of the high frequency harmonics, which means that your brain activity is more free to diverge from the way that your brain is hardwired. It's more free to explore different and diverse patterns of activity than the ones that are just hardwired in the structure of the human brain. And as a way of redescribing this notion of structure and function, which I acknowledge is still abstract so far, you can think of brain structure as the pattern of roads in a city or perhaps in a country. And you can think of brain function, so the sharing of information, as the traffic that takes place over those roads. And in particular, this is actually quite similar to what we do, because on a brain, we have brain regions, which you can think of as different cities. And then you have white matter fibers that physically connect those regions. And those you can think of as the roads in the brain, the highways. And then you can think of brain activity as reflecting traffic, because that's what it is in the end. It is which regions are active more or less at a particular point in time. So just like traffic, brain activity evolves over time. And just like traffic, brain activity is not always the same. It's not always bearing the same relationship with structure. So the plot here is a plot of how much traffic there is over the course of several weeks. And what you can see is that there is quite a striking pattern. During weekdays, when everybody needs to be in the big city, there is a lot of traffic. But during the weekend, when everybody is free to do whatever they want, there is much less traffic. So what you can see from this picture is that even though the roads are exactly the same, so the structure is the same, the relationship of that traffic to the structure, the relationship of function to structure, can be different at different point in time. And that is what we are seeing with these harmonics and what others have shown before. That is to say, under anesthesia, or when you're in a disorder of consciousness, the structure tightly predicts the information sharing in the brain. On the other hand, when you are in a psychedelic state, our recent results have shown that brain structure is less good at predicting what brain function is happening at a particular point in time. There is a more diversity of repertoire. And so once again, we go back to this comparative approach to consciousness. That is to say, we have found some signatures of what the unconscious brain looks like that are not specific to anesthesia or not specific to disorders of consciousness, but rather they are shared by these two different, very different ways of losing consciousness. But this still leaves us with a question, which is, how can such very different ways of losing consciousness result in the same behavioral endpoint of unresponsiveness and result in the same subjective endpoint of loss of consciousness? Because these really are quite different. Disorders of consciousness involve anatomical damage, and they are persistent, chronic. On the other hand, anesthesia, regardless of which anesthetic agent is being used, it is reversible, and it is a pharmacological modulation of consciousness. It's acting on neurochemistry. It's not acting on neuroanatomy. So one of them is permanent, the other is not. One of them acts on function, on structure, the other acts on modulation. How can they result both in loss of consciousness? That was the question that we tried to address. And we took a page from Richard Feynman's book, which is probably something that we thought might be wise to do. And Richard Feynman famously said, "What I cannot create, I do not understand." Now, we are not so grandiose as to think that we can recreate an entire brain and make it conscious, but we think that we might at least be able to recreate some aspects of brain function that we are interested in. And in particular, we take the approach of macroscale abstraction to brain function, which is to say, you can think of the brain as composed of two key ingredients, which is pretty much the two elements of a network. This framework of network neuroscience is relatively recent, but we think it has a lot of power even for exploring consciousness. And indeed, our group and others have already shown the promise of it. So that's going back to the brain. We can think of it as made of these two ingredients: the connectome that I was mentioning earlier, so the pattern of physical connections between each of those brain regions, so-called the cities in your brain, if you will. And at the same time, you have to combine that with the way that each individual region in your brain normally works, which is, in this particular framework, a biophysical model of different populations of neurons that are interacting with each other. So each of these populations of neurons in this particular model is identical, but they are differently connected to each other. And as a result, we can observe some realistic brain dynamics emerging from this relatively simple yet biologically powerful model. Importantly, this model can then be perturbed in each of its two key ingredients. So for instance, you can simulate injury by lesioning the connectome. Of course, we're talking about a virtual lesion of a virtual connectome. I should be clear about that. On the other hand, you can also simulate anesthesia or another kind of pharmacological intervention by acting on the excitation and inhibition that are governing the patterns of neuronal activity in each of these biophysical models that represent how your brain regions work. And you might notice already that the first intervention, acting on the connectome, looks like perhaps what happened to patients with disorders of consciousness. Whereas the second intervention looks like what might be the effects of an anesthetic agent on the brain. And so that is the approach that we took for this. And of course, the model is actually a little more complicated than what I've just been describing. And this is a description of what the full model looks like. This is called a dynamic mean field model. And if you're interested, There is the original description of this approach in this paper by Gustavo Deco and others from 2018. It's a great paper. And actually, it's a paper about LSD. So I hope all of you already know it or will check it out if not. And the description of the particular approach that we took to understand loss of consciousness is in this other bioarchive preprint. So moving on, as I mentioned, we think that these two approaches can represent anesthesia or represent disorders of consciousness, again, virtual anesthesia and virtual disorders of consciousness in our model, in a way that might give us some insight about how, despite their differences, both of them can lead to loss of consciousness. So let's start with the virtual anesthesia. What we did is we took a pattern of how much each brain region is receptive to the effects of this neurotransmitter GABA. Now, the reason we're interested in GABA is because GABA is an inhibitory neurotransmitter, which is the main target of the anesthetic drug that we use, Propofol. We know that Propofol binds to GABA receptors, and we know that that is how it induces loss of consciousness. And luckily enough, thanks to the work of other scientists, we know how responsive each region is to the effects of something like a GABA agonist, like Propofol, because we know how many receptors that region will express. And so we can incorporate this into our model, and we can ask the following question. If we are taking into account how much each region will be responsive to GABA, can we predict what is going to happen when we increase the inhibition? And the answer is yes. That is to say, on the plot on the right here, you can see how well our model fits empirical data. Now, one aspect that I should emphasize is that in this particular plot, higher is worse, because what is being plotted is the divergence from a real functional MRI dataset. So remember, what we're doing is we are simulating functional MRI data with our model. And then the question that we're asking is, how similar is this to real data from awake conscious people, or to real data from anesthetized people, or real data from our DOC patients? So lower means less distance from the real data. So it means a better fit of the model. And we can see that when we simulate anesthesia, in both cases, we get not only a better fit to the real anesthesia data, which is nice confirmation, but we also get a better fit to the patient's data. So these are patients that are not under anesthesia, but they are unconscious because of some brain injury. And nevertheless, by simulating what happens to a brain that is under the effects of inhibition, simulating anesthesia, we can also simulate the brains of DOC patients. And then we took the opposite approach, which is to say, we try to simulate what happens to the model when we replace its healthy original connectome with a connectome taken from these DOC patients, for whom we actually already have the data about the way that their brains is wired. And we know that that is significantly differently wired than the brains of healthy individuals who are conscious. And once again, we use both the empirical data from anesthesia and the empirical data from patients with disorders of consciousness to identify how good our model is at recapturing what is really happening in those brains. And once again, we find that with the model that is using a lesioned connectome, we replicate not only the data from the lesioned, brain lesioned patients, but we also replicate the data from anesthesia. And of course, what this does not tell us is that anesthesia is also rewiring your brain. That is not happening, and we know that it is not. What it does tell us is that in some sense, the kind of brain rewiring that these patients have undergone is functionally equivalent to the kind of increased inhibition that is produced by Propofol. So even though they have very different mechanisms of action on neuroanatomy and on neuromodulation respectively, their functional effects can be demonstrated to be equivalent. And this is what seems to lead to unconsciousness in both of these cases. So I'm going to wrap up now, and I hope what you can take away from this is that we are starting to have the tools to map a landscape of consciousness based on brain function. And by comparing the brains of different kinds of unconscious individuals, we can understand where they stand with respect to the healthy brain, and also where they stand with respect to the brain that is under the effects of psychedelics. So bringing all of these different perspectives together as a way to understand what consciousness is, and maybe even one day how it can be restored in patients who have lost it. And this is just an overview of some of this research, trying to bring it in context. And the approach that we are taking is what we like to call a synergistic cycle, whereby we start from brain data and we turn brains into networks using tools from graph theory and network science, because that lets us obtain insights from the mathematics of graph theory and network science, which are well developed and well established. And once we have those insights on the brain as a network, we can then use machine learning and the kind of brain modeling that I've been describing to translate those network insights back into insights about the brain. And of course, this is not work that I carried out on my own. This work was the result of extensive collaboration with very many people. And there are actually many more than these. These are just the people who are involved directly with the studies I've been talking to you about. But in fact, this list should go on even further if I were to describe all the collaborators from all of the studies that we have done. And dates then not just to the University of Cambridge, and in particular, the Cognition and Consciousness Imaging Group and my wonderful lab members, and my advisor, Professor David Menon, and my supervisor, Dr. Emmanuel Stamatakis. But also we have collaborators in many other institutions, and here shown are our collaborators from Oxford, from Imperial College in London, and from Trinity College Dublin and London, Ontario, who have all contributed data or analytic resources or their insights into making these projects possible. And finally, I should also express my gratitude for Gates Cambridge, who are sponsoring my PhD, and thanks to whom all of my research is available open access for anybody to read, which I think is very important. And finally, thank you guys for your attention. And I would be happy to answer any questions that you may have.
All right, great, Andrea. Thank you for giving this talk. Yeah, I would just let's just go with questions. So you can stop share, or no, you can keep sharing in case we need to go to the slides. And yeah, if you have questions, maybe raise your hand or in virtually, or just start talking. And yeah, please go ahead. Otherwise, I'm gonna ask some questions.
Yeah, I would have a question.
Sure.
So yeah, I recall in 2019 there was already talk of giving psychedelics to coma patients. So it seems like this was this sort of disparity was already noticed, and I believe the coma science group in Belgium is also have been sort of talking about doing something like that. I'm wondering if your group is currently planning anything like that, and whether you've maybe used any of these modeling approaches to predict maybe which patient might be most benefiting from an intervention like that, or if you can use modeling to make predictions like that in the current
Sure.
state of the art.
Yeah, so the latter is the question that I'm closer to being able to give an answer to, which is to say that yes, we are very interested and we are actively looking at different kinds of models that we can use to predict whether someone might benefit from different kinds of interventions, potentially including pharmacological interventions, in terms of restoring their consciousness. Because of course, for each of these patients, we have their individual connectomes. Everything that I've been showing is based on an average connectome from these patients, mostly for computational reasons. But in fact, we have each of their individual connectomes. And of course, the challenge is that they are quite different. And we know that some patients can benefit from different kinds of pharmacological interventions than others. So there are some patients, paradoxically, who benefit from zolpidem, which is a GABAergic agent, and it is usually used to help treat insomnia. Other patients do not benefit from that, but they seem to benefit from dopaminergic agents instead. So there are trials going on about amantadine and other different kinds of drugs. So it is an ongoing challenge, and that is something that we are very interested in pursuing. Of course, there are very serious ethical questions with respect. Psychedelic research comes with a lot of ethical restrictions because first, they involve typically restricted substances, and second, we want to really be sure that the volunteers are going to be safe. So there is a lot of, each of these studies underwent very rigorous ethical approval procedures before being approved. But of course, studies with patients with disorders of consciousness, especially if they are clinical trials, also involve severe ethical procedures to make sure that the patients are being, their well-being is being safeguarded properly. So combining the two of them is especially challenging. And I can tell you that not everybody, even who is working in these fields, is on board with this kind of idea. So I don't know to what extent the two of them will be combined soon. But I definitely can tell you that we are in fact pursuing using the kind of modeling that I've been describing to identify what could happen.
Yeah, do you know if Ambien, or that's the other name of it, I believe, does that have these same harmonic signatures as psychedelics, for example? Like, does that?
We do not know.
Mm.
I am not aware of anybody having tested that. It is something that I would very much like to test.
Yeah, because with that one, we know that it does help the coma patients, but we don't know. Then that would be nice, right?
Yeah, there are many, and likewise for these dopaminergic agents like amantadine, we do not know yet what their harmonic signatures look like. But would be very nice to look at that. Anyone else?
I can ask a question.
Sure.
So I was wondering if you had looked at what happens on anesthetic doses of ketamine. And also I wondered, I've seen in the medical literature that ketamine and propofol are often administered together for anesthetic purposes. And I think in that case, it's roughly a sub-anesthetic dose of ketamine. And I wondered what the dynamics underlying that were and if they'd been studied too.
Right. So the first, unfortunately, the answer to both of those questions is no, which is to say we would love to have access to a ketamine, anesthetic levels of ketamine with fMRI data associated that we could actually study to identify the signatures. Because of course, that is perhaps the crux experiment to do in terms of determining, is this about the molecule, in which case it should look like the sub-anesthetic, or is it about the state of consciousness, in which case it should look like propofol, despite being a different kind of drug. So we would love to look at that. We typically tend to study individual drugs because often their interactions are very complex. And then attributing back what we are observing to each individual drug or to their interaction would become a much more complicated matter, especially given that we don't fully understand yet the effects of either of those on the brain, as the fact your previous question was just revealing the limits of what we know so far. I think, and again, I should come clean at this point. My background is in philosophy. It's not in medical science. I do not have a medical degree of any sorts. I'm not even a doctor yet in the sense of PhD. But given that, I think possibly why propofol and ketamine are used together may be to help with the breathing suppression that happens at high levels of propofol, which I do not think ketamine induces. So combining them might alleviate that potential problem. But this is only my hunch. from what I've read about each of them.
Okay, interesting. Yeah, I've looked a little bit about the relationship between ketamine and the perturbational complexity index, and the results were not so clear then between the sub-anesthetic and the anesthetic dose. And it makes you wonder if people under the anesthetic dose are truly unconscious or not, and it's kind of a difficult question to answer. So
it is even more so because ketamine, even the so-called anesthetic levels of ketamine, ketamine is a different anesthetic from the others because it is a dissociative anesthetic. So sometimes it can be used in a way that doesn't induce unconsciousness, but it induces a dissociative state, whereby people still say that they perceive the pain, but not as painful, and they don't care about it, in which case they can still be operated on, despite not being, as we would say, unconscious, because they still have some mental subjective experiences going on. So ketamine is a very interesting drug, but also as a result, or perhaps as a reason, very messy study.
Okay, thank you.
Sure. Anyone else?
There's a question in the chat.
Can't see the chat right now.
Is there data on connectome harmonic decomposition in REM sleep? If so, how does this compare to an awake state?
Again, I do not think that there is data on REM sleep yet. This I should add, this is a very recent technique. So even our work, it's available as a preprint, but it's not even published yet. So at the moment, the only studies that I'm aware of as being officially published are the ones on LSD and psilocybin that I have mentioned here. I do know that there is ongoing work on non-REM sleep and on meditation by Selen Atasoy and her colleagues. But for most other states that you can think of, the answer at the moment is going to be, I don't know, but I would love to find out.
Yeah, I have one question.
Sure.
So we talked about the connectome. We talked most about the macroscopic connectome. At the modeling level, then when it comes to the connectome harmonics is a bit more of a mesoscopic connectome. I guess one question that I have would be like, because there are two factors at plays in structure, right? There is a white matter, there is geometry of the cortex as well. So I would like to know if you if you think how those two balance. So the geometry of the cortical surface with the long range connections from white matter in terms of these decompositions.
Yeah. So that is a very good question, because it is true that the kind of generative modeling that we use does not actually include the local gray matter connection between nearby regions, but it models each region as a discrete entity that is only connected to the others through these white matter fibers. And that is the approach that is most commonly taken for this. But we actually do know that both of them matter for reconstructing the empirical connectome harmonics specifically, as Marco knows well. And so that is something that definitely I think should be looked into more. So I think that is why I was mentioning specifically not just macroscale, but macroscale abstraction, because there are many different levels of complexity or biological detail that we have been abstracting away to make this particular kind of model tractable. So for instance, one reason is we need to reduce the brain to a number of regions. And of course, it might be surprising for those of you who are not neuroscientists, but there isn't actually a single agreement on what counts as a brain region. Pretty much everybody and their grandmother has a parcellation of the brain, that is to say, a particular way of identifying what counts as a brain region. Some of them are very big regions, some of them are very small regions. So for this analysis, we had, when I was mentioning the whole brain models, we had 68 regions. But there are alternative views that view the brain as divided into up to thousands of regions. So that is another question that will need to be addressed. And of course, that bears different importance for the question that Marco just asked, because if you only have these very macroscopic regions, that maybe everything within a region is already counted as connected together, and the only things that really matter at that point are the long-range connections. Whereas if you have very many small regions, that maybe that is failing to capture a lot of long-range connections, because actually they should be locally connected as well. So those are a lot of different aspects that interact with each other. And in particular, there are also different kinds of models themselves. So the model I've been presenting has these two ingredients, which is the connectome. As Marco was just mentioning, there are multiple ways of identifying what counts as a connection between different regions. But as I mentioned, the other ingredient was this biophysical model. And we use this model of interacting neural populations of excitatory and inhibitory neurons. But there are other models that don't include these biophysical aspects, that just, for instance, the Hopf model or the Kuramoto model, they just look, well, they don't just, but they look at brain regions in terms of oscillators, effectively. So they abstract away some of the extra neuronal complexity that the models that we have been using try to keep instead. And as a result, they also gain some computational properties such as scalability. So there is always this trade-off in computational modeling. And in some sense, it's about getting the kind of complexity that we need to understand the questions that we want to understand. I know that is in some sense not the most satisfying answer. I wish I could say the answer, I wish I could say as an answer, we're going to model everything.
Yeah, but
and I think there are people in Switzerland who are trying that, and I think they have been trying for the last 10 years.
Yeah.
So
I think it may not be the most satisfying answer, but I think it's the best one that we have. It's actually in the sense that, I think, as you were saying, there's different scales of modeling, there's different approaches of modeling. And I think the point is just to understand what the question is, right? And then whatever your model or approach is, it has to be tailored to, you have to show that it's the right approach to answer a specific question. There isn't one approach that is right and other approaches that are wrong. And there's a lot of insights, of course, that you can gain about the brain at all these different scales, from the more microscopic to the more macroscopic. So I think that's also why what's so interesting about this field right now. So
yeah,
I think that's the best possible answer that you.
And I should actually add about that this whole brain, this kind of dynamic mean field model that we use is not the only one that has been already applied to obtain insights about consciousness. So the other models that I was mentioning, the Hopf model, but also the Kuramoto model, the Ising model, all of them have been used in the last couple of years and are being used to identify relevant aspects of consciousness. So this is not just the standard way to do things. All of these approaches are, I wouldn't say even contenders, because they are not contending with each other. They are complementary to each other. They're using different avenues to identify and resolve different questions.
Yes. Any more questions? I have another one then, because we started from. Was there a question? No. So in the very beginning, you were showing the specific region that was in common between the different modes of loss of consciousness. And some months ago we had another. Do you remember what the name of that region is, or like what is it?
Yeah, so that is the posterior cingulate / precuneus. It's a bit at the border of the two of them.
All right. I was
sorry, no, go ahead.
Some months ago we had the journal club. There was a paper about ketamine and the retrosplenial cortex, I thought it was near there. I'm not a neuroanatomist, but yeah. Is it nearby? So do you think is that the same thing? Do you think it is the same thing that you're seeing with that approach as they've seen in mice and with ketamine?
So it is definitely the region that I'm talking about is definitely very close to retrosplenial cortex in the human. Now, that region is also among the regions that have undergone the most level of evolutionary expansion, even from chimpanzees or macaques to humans. So it is not straightforward to say whether it is the same region as in the mouse in that sense. But I do find it very striking that it is, at least on a neuroanatomical mapping level, very closely related. And I think you're referring to the Nature paper from a while back. This is very exciting. I think the convergence that we were seeing is really quite striking. I should say the same region is implicated in a lot of different kinds of studies. So it is one of those regions that also becomes sort of disintegrated, as the term is, during the experience induced by LSD. So that region is coming up over and over and over again. And we have recently some work in the lab as well that is showing what happens to that region as you lose consciousness in terms of its connectivity with some neuromodulatory systems. So that seems to be, in some sense, almost like a light switch when it comes to the brain. Lots of things in the brain seems to depend on it, and in turn it seems to be implicated in a lot of different occasions where brain activity and brain function become very substantially altered, from disorders of consciousness and anesthesia to LSD and other psychedelics.
All right, thank you. So there is a question in the chat. Can you see the chat, or should I read it out? Or maybe I should read it out loud for the recording as well. So the question is from Alexander. How do connectome harmonics based on fMRI relate to the frequency bands measured by EEG, MEG? As far as I understood, the connectome is based on the temporal frequencies of activity patterns as picked up by fMRI, while the other depends on electrical oscillations that seem to organize neuronal activity. To what extent does this resemble each other?
So one point that I need to clarify is that in this case, the connectome that we are talking about is not the functional connectome. So there is a structural and a functional connectome. The functional connectome is what I was describing as the traffic, which regions are active together over time. The structural connectome is the roads, which regions are physically connected. And that is the one that we are using for our decomposition. There is currently a preprint by, again, Atasoy and colleagues that is looking at a functional harmonics decomposition, but that is not what we did in this particular case. Now, in terms of how they relate to the EEG, the answer is, unfortunately, again, we don't know. So there are people in, I think, Switzerland who have been using a process similar to connectome harmonic decomposition, the difference being that it doesn't include these local connections that Marco was mentioning, to look at EEG signals. So we are starting to build up towards that. But at the moment, when we talk about frequencies in terms of connectome harmonics, we can only talk about spatial frequencies. So how coarse-grained or fine-grained a particular harmonic pattern is. We do not yet know how that relates to the frequency, the temporal frequencies that are typically captured by EEG. And again, the reason is not that we're not interested, because we are, but as I think just from the questions that have come from this chat have shown, is that there are too many questions to be answered all together. And so we are trying to pick the ones that we think are going to be the most helpful for building the kind of science that we're doing next. So I am expecting that in the next 5 to 10 years, most of these questions will have been addressed. Some of them we are working to address right now, but it is a very, very recent field of study. And that comes with, which is good for us, lots of unknowns yet to be solved.
All right. So thank you, Alexander, also for the question. So I think if there are no further questions, I would be inclined to end it here on that note, on that optimistic note for the future. I think there's lots of questions to be asked and partial answers to be found. Probably no complete definitive answers of how the brain works anytime soon, but you know, some probably some good answers, as we have already seen some, I think, in this presentation.
And yeah, thank you so much for having me. It was a pleasure talking to you guys and having such an engaged audience. I've just added in the chat my email, so that if you want to reach out with any further questions, I'll be very happy for you to do so. Just drop me an email, and I'll be happy to interact with any of you.